Good day everyone. My name is Jane Ray H. Torion and we're going to tackle about tissue culture along with my group mates. What is tissue culture? Tissue culture is a process that uses plant material in a growing medium to grow new plantlets. The initial plant material is cultured and developed in a specific and tightly controlled environment. So tissue culture no, is a technique of instead of planting the seed in soil, you can cultivate a tissue, organ, or cell into a specially formulated nutrient media, a sterile condition, and controlled environment. And by it can create a new whole plant. So, 1902, Gottlieb Haberland proposed the theoretical basis of plant tissue culture. He is known as the father of plant tissue culture. He experimented with isolated photosynthetic leaf cells but wala siya na successful in inducing any growth. Wala siya not as successful because the plant that he experimented did not multiply. However, he predicted that one can obtain artificial embryos from vegetative cells using this culturing technique and established the concept of totipotency. So the idea of totipotency is discovered by Gottlieb Haberland. So in 1907 to 1910s, first practical cultured, Ross Harrison and Alexis Carrel developed the first successful in vitro animal tissue culture, establishing aseptic technique. So Rose Granville Harrison was a American biologist, and he is known as the father of animal cell culture because he developed the hanging drop method to observe nerve cell growth. So he was the first person to successfully grow animal cells outside the body. So in 1907, he grew cells from frog embryos in glass dish using a simple medium. So he discovered that nerve cells can grow and develop outside the organism. He showed that cell can survive, divide, and form long nerve fibers in a controlled environment. While Alex Carrel was a French surgeon and a biologist, and he won the Nobel Prize on Medicine in 1912 for his work on blood vessels, and organ transplantation. So the work of Harrison, he improved the technique for growing tissues outside the body. He created the special sterile method to keep tissues alive for long years. So he grew pieces of chicken heart tissue and maintained them for many years. He showed that animal tissues can stay alive in a nutrient solution if the environment is clean and controlled. So he developed the Cargill flask, a special bottle for tissue culture. His research helped the scientists understand how to keep the tissues in sterile and nourished. So also he coined the word tissue culture together with his assistant Montrose Burroughs. So in 1930s to 1950s, explant and callus studies by Roger Gutheret, Pierre Nobicourt, and Philip R. White, researchers advanced aseptic plant, explant, embryo, and callus culture methods, setting the groundwork for modern protocols. So Roger Gutheret is a French plant scientist and one of the father of plant tissue culture. He experimented with growing tiny pieces of plant tissue, especially carrot tissue, in a nutrient solution inside the laboratory. He studied how plant cells can survive, divide, and grow outside the whole plant. So his discovery in 1939, Gutheret successfully grow plant tissues continuously in a vitro or like in test tubes. He discovered that plant cells can keep dividing and forming new tissue when placed in a right medium, and this proved that plant cells have the ability to grow independently, a major foundation of modern plant biotechnology, which is the tissue culture. While Pierre Nobicourt, a French botanist and another important contributor to early plant tissue culture research, he improved nutrient media used for growing plant tissues. He studied how plant parts respond to different growth substances. So he also worked on callus culture, the mass of growing plant cells. So Nobicourt discovered that specific nutrients and hormones are required for a plant tissues to grow well. So experiment helped identify which minerals, vitamins, and growth regulators improve plant cell growth rather. And lastly is Philip R. White is an American scientist known for pioneering root culture. He grew isolated tomato roots in test tubes and kept them alive for many years. So he created nutrient solution that supported long-term plant tissue growth. And White discovered that plant root plant roots can live and grow independently when given the right nutrients. So he showed that it is possible to keep plant tissues alive for a long time outside the plant body, and the nutrient media become the basis for the future plant culture techniques. So the first basic principle of tissue culture is totipotency, and it means that a single plant cell can grow into a whole new plant. So even a tiny part of a plant like our leaf or root cell has all the instruction needed to make the entire plant. So if you place that cell in the right environment, it can grow into a full plant. And this idea was discovered by Gottlieb Haberland. The second basic principle of tissue culture is aseptic techniques. So in plant tissue culture, keeping everything sterile is crucial because plants, like any living things, can get infected easily by bacteria, fungi, or other microorganisms that can harm them or stop them growing. So aseptic techniques are the step taken to make sure that the environment is free of these harmful microorganisms. So the basic techniques are sterilizing the tools that you're gonna be using when performing the experiment, and also sterilizing the plant materials for to avoid contamination and also to clean work area. Also, last um, basic principle in tissue culture is controlled condition. No, when growing plants in lab, the environment must be controlled to make sure that plant cells grow properly. This means carefully adjusting things like temperature, light, and humidity to give the cells the best chance of growing into a healthy plant. So next is the types of tissue culture technique. First is the callus culture. A callus refers to a cluster of undifferentiated cells with a remarkable ability to give rise to various plants. So a callus no is a mass of soft, unorganized cells like a lump on of a plant tissue, uh, which is a piece or like leaf or stem or root is placed on a nutrient medium with hormones. It forms a callus. It's called callus. This callus can later grow into a root or shoots or even a whole plant. So example for the callus culture is used in sugarcane to produce many new plants for like farming. Second is the seed culture. Seed culture is a form of tissue culture widely utilized in the cultivation of various plants, including orchids, in a lab environment using seed as an explant. Seed culture no uses obviously seed as the starting plant or like explant, and grow it into the lab under a sterile condition in nutrient medium, and it is useful for plants that are it is useful for the plants that has a very small seeds or like hard to grow in soil. And the example next is the protoplast culture. A protoplast is a plant cell lacking on a cell wall. In this technique, the cell wall of the plant cells is eliminated through mechanical or enzymatic means. So protoplast no is like a plant cell without its protective cell wall. No, it the cell wall is removed using enzymes, then grow the naked cell and make it into a whole new plant. And this protoplast can be fused with other to combine traits of two plants. Example is creating a hybrid of potato and tomato by fusing protoplast by using it. In this technique, you're going to use anther or like pollen, pollen grains or whole anther, the male flower parts are cultured, no, they can grow into haploid plants or like plants with one only set of chromosomes. So haploid plants are very useful in plant breeding because they can quickly be converted to pure lines. So in ovariculture, this technique we're going to use fertilized or unfertilized ovary as the explant in and put it into a nutrient medium and helps in developing hybrid or when natural fertilization is difficult.
So let's talk about the materials, equipments, and procedures used in tissue culture. First is the materials. So sucrose is a simple sugar is added to the medium as a source of energy because the plant can't produce or can't get enough food through photosynthesis while in tissue culture. Next is agar or gelling agents. We mix agar into a medium to make it semi-solid so that the explants stay in place and can absorb nutrients easily. And plant growth regulators, these are plant hormones such as auxins and cytokinins. They control cell division and tell the tissues when to form roots, shoots, or callus. Plant explants, these are small pieces of plants like a leaf segment, like a shoot tip that we use to start a culture with. And they contain living cells capable of developing into a whole new plant. Sterilizing agents, chemical like 70% ethanol and sodium hypochlorite help sterilize the plant tissues, the tools, the workplace to prevent bacteria and fungi from growing. Next is the equipment used for tissue culture. First is laminar airflow cabinet. This is a sterile workstation that blows clean, filtered air to keep the area free from contamination while handling plant tissue or media. Next is autoclave or autoclave. We use an autoclave to sterilize everything, like media, tools, containers, and using hot steam under pressure. It kills bacteria and fungi and spores. Third is Next is the growth chamber or incubator. This machine gives us the right temperature, humidity, and light cycle to help our tissues grow properly, like simulating day and night. Number four is microscope. We use a microscope to check the growth of cells and detect any contamination that you can't see with the naked eye. Five is pH meter. Before we sterilize our culture media, we use a pH meter to make sure it is the right pH level. So water purification system. Clean water is very important. We use purified or distilled water in all our media and chemical preparations to avoid contamination. Next is shaker. When growing cells in liquid culture, a shaker keeps them moving so they can get enough oxygen and nutrients evenly. And glassware and plasticware. These are the test tubes, jars, or petri dishes where we grow our tissue in nutrient-rich media under sterile conditions. 9 is refrigerator. We store chemicals, growth regulators, and prepared media here to keep them stable and effective. 10 is hot air oven. This oven uses dry heat to sterilize glassware and metal instruments at high temperatures. So let's proceed to general procedure. First is selection of explant. We select part of a plant like a leaf, root, or shoot tip known as explant. Second, surface sterilization. We sterilize the explant to remove any microbes on the surface. Third, preparation of culture medium. We prepare nutrient-rich medium that supports plant tissue growth. Fourth is inoculation of explant. Now we place the sterilized explant into the medium in a sterile container. 5. Incubation in growth chamber. We incubate the culture in a growth chamber under controlled conditions. 6 is callus formation or organogenesis. Depending on the hormones in the medium, the explants forms either a callus, shoots, or roots. 7 is subculturing. To keep the tissues growing, we transfer them into a fresh media regularly. 8 is rooting. If only shoots have formed, we transfer them into a medium with rooting hormones. 9 is hardening or acclimatization. Finally, we move on the plantlets to soil or potting mix gradually to adjust to the outside conditions.
Okay, let's continue the application in plants. First is mass production of disease-free plants, means growing a lots of plants with a free virus. Um, Use meristem culture, which involves growing the tip of a shoot that naturally virus-free. Uh, Meristem culture, a method where the scientists take a tiny tip of a plant shoot, which is naturally free of viruses, and growing growing it in a lab condition to produce clean and healthy plant. Okay, next is crop and genetic engineering and use special lab techniques to make plants better. Um, Tissue culture allows for precise introduction of desirable genes. Um, Tissue culture, where the scientists grow plant cells in controlled condition and add specific genes, like ones that help plants survives of resist pests. Um, this help make create strong and uh, healthier crops with desirable traits. Next is a um, 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 conservation of endangered species. Um, Means protecting rare plants from extinction. Um, Rare or endangered plants can be propagated without disturbing their natural habitat. Um, using culture, using tissue culture, scientists can grow the plants in labs, starting from a few cells, or, for example, the tiny, tiny, uh, tiny parts plants of the plants, without needing to take them from the wild. This help preserve their natural habitat while increasing their numbers. Uh, Next is a production of secondary metabolites. Um, Means making useful plants chemical in labs. Um, Culture plants can produce a valuable compounds like alkaloids, flavonoids, and terpenoids. First is a alkaloids is used for in medicine or painkillers, and next is a flavonoid is a used for a antioxidant. We get this in fruit. Next is terpenoids for a perfume or essential oil. So we need to protect our plants because we can be. Next is a prospect bioreactor system for large scale control. High likely technique growing plant in machine. Bioreactor offer control environmental, for example, right temperature, nutrients, and oxygen, and growing plant cells, tissue, and organs in large quantities. This is a useful for producing a multiplying efficiently um, at a industrial scale. Is synthetic seeds um, created by scopulating somatic embryos or shoot buds in a gel-like coating. Why we need to coat the shoot buds? It because to help the plant a fast growth and make it easy handle and just like a normal seeds. Um, Organoids and regenerative medicine implants a cutting edge area of research. Um, though more common in animal system, plants organoids miniature organs are emerging for studying development and stress response. Nowadays, not only for animals, but the scientists develop for plants also because study how plants grow and develop, understand how they respond to stress like diseases. Okay, CRISPR-Cas9 and gene editing with tissue culture. Best combo for improving plants. Um, Tissue culture is essential for regenerating whole plants or edited cells. Uh, Precise tool that edit specific genes in plants. For example, delete the bad genes and add the good one. And lastly, the conclusion. Tissue culture enable a rapid plants propagation, genetic preservation, and medical research. It supports food security, advanced medicine, and conserve biodiversity. As a versatile tool, it addresses global changes and fosters sustainable solutions for agriculture, healthcare, and environmental conservation.